over to our next speaker, who is Katie Warnaby, who is a senior research scientist at the Wellcome Centre for Integrative Neuroimaging at the University of Oxford. And she will talk about measuring depth of anesthesia. Over to you, Katie.
Whoops, sorry, wrong one. Hi there. Let me just sort this out. There we go. Thank you, Andre, and thank you very much for the opportunity to speak today. So, as Andre said, I'm a researcher at the University of Oxford, and my interest is using brain imaging to try and understand depth of anesthesia. And so I'm really trying to address some of the fundamental outstanding questions in anesthesia. And they are, how do anesthetics produce loss of consciousness? How do they affect awareness and the perception of external stimuli? And I hope that, you know, through this improved understanding, we can try and develop some better clinical measures for depth of anesthesia. And that can potentially lead to optimizing dosing for each individual during surgery. So general anesthesia is really a cocktail of drugs that are given to prevent awareness, prevent pain, and to stop people moving so surgery can take place safely. And as we increase the concentration of an anesthetic drug, we take somebody from a state of alert wakefulness to deep unconsciousness, whatever that may be. And at some point in that continuum, as we've discussed, they will lose responsiveness. But then they enter into an indeterminate state, really, where we're not sure what they can feel, what they can perceive, whether they can hear. And this really was sort of our first question, is when does an individual stop perceiving the outside world? So clinically, this is a problem. As Johan said earlier on, there are 234 million general anesthetics delivered daily worldwide. So, you know, there are a huge number of surgeries taking place every day. And the anaesthetists who deliver the anesthesia for these surgeries have a delicate balance to obtain. So they want to give you just enough anesthesia so that you're not aware during surgery and you don't feel any pain. But they don't want to over-anaesthetize you either, so that your physiology, your heart and lungs are impaired, or it takes you longer to recover. And particularly now, there's increasing evidence that over-anaesthesia, particularly in the elderly and vulnerable, may have sort of both short-term and long-term influences, such as delirium. So currently, what anaesthetists do is they will say, right, let's look at the general population and say, for your age and for your weight, this is the dose I should give you. And then I'll probably give you a little bit more just to ensure there's a safety margin. And then I'll monitor your heart and your lungs, and I'll see if there are any changes when, you know, the surgeon puts in some stimulation, he's moving things. And one of the things that I guess surprised me as a physicist and neuroscientist is it's very rarely that anaesthetists actually monitor the brain. So there was a survey done in the UK, and only 2% of anaesthetists would regularly, on a case-by-case basis, monitor the brain during anaesthesia. And I, you know, find that very strange, seeing as it's the site of consciousness. And I guess the reason for this is the current brain monitoring that we have to monitor depth of anesthesia is really not very good, and sometimes doesn't even monitor responsiveness very well. And so what we really need is a reliable and individualized target that tells us when people are no longer perceived under anesthesia. So you've already heard about some of these techniques from Olivia. I'm based in the Wellcome Centre for Integrative Neuroimaging in Oxford, and we have a number of magnetic resonance imaging techniques at our disposal. So we can put people in a scanner while they have anesthesia. And one of the ones I'm going to be talking to you about today is task MRI. So I can deliver a number of different stimuli to my healthy volunteers. I can deliver pain, I can give them beeps, I can deliver words, and I can see how their brain changes under anesthesia. We can also look at people's resting activity of their brain and look at how that changes when they're in an anaesthetised state. Olivia also showed some DTI data earlier on. It may be that the strength of connections, the strength of wiring may relate to anaesthetic susceptibility, why some people need more anaesthesia than others. And we can also segment the brain using volumetric measures. So we can look at the amount of white matter, the amount of grey matter people have. And again, you know, the volume sizes of certain regions of the brain may relate to susceptibility. And finally, when we give anaesthesia, we're ultimately altering the brain chemistry. And we can use techniques such as magnetic resonance spectroscopy to look at how these drugs influence neurotransmitter levels in the brain and in specific regions. So we performed a new set of experiments. The results aren't quite out yet, but we've incorporated all these techniques. So hopefully we'll be able to provide you more information about how these change your anaesthesia in the near future. So another technique that Olivia's alluded to already is EEG. So this is where we measure the electrical activity of the brain. And it's used very often in sleep, and I'm sure a lot of you are aware of REM sleep and slow wave sleep, the early phases. And actually it's these electrical signals that are used to define these different sleep stages. And effectively what we're doing is we can take a signal like this and we can decompose it into different frequency bands. So this one here would be an example of a low frequency with a higher frequency signal superimposed and then a medium frequency one. So that will come in later when I show you some results. So one of the things that my lab does is we combine EEG and MRI. And whilst this is really technically challenging, it does have the advantage that we get the best of both worlds. So EEG, electroencephalography, has really high temporal resolution. I can say when things are happening in the brain with millisecond resolution. With fMRI, I can tell you where things are happening. So combining the two, we can get a good picture of anaesthesia and how it changes the brain. So I'm going to talk to you and take you through a discovery that we found of what we think may be a new marker of depth of anaesthesia. And this came out of some experiments quite a long time ago now, I guess 10 years ago when we did the actual experiments, where we put healthy volunteers to sleep really slowly. And we did two experiments. We did an EEG experiment and then we did an EEG fMRI experiment. And what we did was we delivered the anaesthesia incredibly slowly. So we took what happens clinically in about 10 seconds and we spread it out over the course of about 50 minutes. And this allowed us to use the imaging to really watch the brain in slow motion as it switched off, and then as it re-emerged, as consciousness re-emerged in these individuals. So we, first of all, held people at rest with their eyes shut for 10 minutes, and then we increased the dose of propofol up to a maximum level and held them again for 10 minutes. Then we switched the drug off and we watched them emerge from anaesthesia. So during these phases here, phase 2 and phase 4, during the induction and emergence, we also gave them lots of stimuli. So I gave them words. We used lasers to apply to the lower leg to give painful sensations. It heats the tissue up. It feels like a bit of a pinprick. And then we also got them to perform an auditory word task. So we gave them pairs of words. So for example, I would present to them duck and ball through headphones. And we would ask them to press one button if the words were the same and a different button if the words were different. By using this particular task, we could identify the precise point where each individual lost responsiveness under anaesthesia and gained responsiveness on wake up. So here I'm showing you the results from the word task experiment. So looking at behavioural responsiveness. And what I really want you to take away from this slide is these are a number of people that are responding at any given time during the experiment. And what we find is some people lost responsiveness quite early, whereas other people were sort of, kept on fighting on to the end and only lost responsiveness very late. And it was the same on recovery. And I think this slide is to highlight the differences between individuals in their susceptibility to anaesthesia. You know, these people here need very little to go off to sleep. These people need quite a bit more. And then we looked at their EEG. So this is, I always think this is a beautiful picture, but basically what we're looking at here is the average result across all individuals, across all electrodes over the brain, for how the electrical activity of the brain changes over time. So on our x-axis, we've got time. The dotted line shows us the propofol dose and how it changes with time. And then on the y-axis, we have activity. So the frequency of these waves that are being produced and how they alter with anaesthesia effectively. And the colour chart shows us really, so when we get to these higher doses, we saw big increases in slow wave activity down here around 2 Hertz, below 2 Hertz, and at these higher levels of alpha activity. So, I mean, this was quite a novel result at the time, and what we were particularly interested in was in this slow wave activity band. And the reason we were interested in that was because there were a lot of there's neurophysiological evidence already that when cortical neurons, and particularly some measurements in the cat, when they were exposed to sort of these hyperpolarizing influences such as anesthesia or sleep, they tended to oscillate between these on and off states, and that's what produces the slow oscillation that we were measuring at the cortex, at the scalp. And we also know that slow activity, you know, is a really key feature and actually is used to define deep sleep. And we know that in those deep sleep phases, that there is a reduced ability to respond to stimuli. So at this point, we were like, well, this is cool. Could this mean that slow activity really reflects isolation from the external world under anesthesia? But one of the key things we realized at this point in time was it's really important to include in our analysis the points where people lost responsiveness, because we couldn't just average over certain drug, you know, concentrations. So what we did is we sort of, you know, we brought all the data together, but we anchored it at where each individual lost and recovered responsiveness. And when we did that, we saw that basically the slow waves started increasing around the point of loss of responsiveness. And then they seemed to reach a plateau later on. Then, when the anesthetic dose was turned off, they slowly decayed. And then when they'd pretty much disappeared, that was when individuals on average recovered responsiveness. And we found that this, what we defined as slow wave activity saturation, seemed to be very prefrontal in its dominance. And we thought, well, this looks really interesting, okay? What we've seemed to have identified is a separate transition in the EEG, so something distinct from behavior. But, you know, do we see this in everybody? We found it in a group result. Can we see it in everybody? So what I'm showing you here is the individual traces. So the red is slow wave activity, the black is the propofol dose, and the blue lines are where people lose and recover responsiveness. And hopefully what you can see is that, first of all, the red lines have different heights. So people can mount this response to a greater or lesser extent. But a key thing in most individuals is that this plateau is reached before the drug dose had finished rising. And this was, but in all cases, pretty much these slow waves only started when they just lost responsiveness, but plateaued later on before this maximal. So what it seemed to us here was that we'd found actually quite an individualized transition as well past this loss of behavior. So we looked then at the ability to mount this response, and we knew it was highly prefrontal in terms of its sort of predominance. And we said that, okay, so if this oscillation really arises from the number of cortical neurons undergoing this particular oscillation, we should see a correlation between that and the amount of grey matter volume in the prefrontal cortex, which is indeed what we saw. So this led us to think, well, actually, maybe SWOS is a potential individualized marker for depth of anesthesia. So we then went into we had the other experiment where we'd done both EEG and fMRI in the scanner. So we then basically saw the same change in EEG. So the slow wave started to increase when people lost responsiveness and reached a plateau later on in general. We could then use the fMRI to look at the brain's response to stimulation around this point. So this top line shows us the responsiveness to laser, so to pain, whereas this one in blue shows us the auditory response. So when people became unresponsive, we saw a decrease in these regions of the brain responsible for processing stimulation. But actually, it was only when we got to the point of slow wave activity saturation that we saw a completely different network that was active and activated by these stimuli. And so it seemed to us, you know, based on the work of Olivia and others in disorders of consciousness, that maybe what was being activated was an internal monitoring network. And when we looked at, we could look at what was more active. If we looked just at that unresponsive period, we could look at what was more active before SWOS than at SWOS. And we found that actually we saw disruption in these thalamocortical circuits that were responsible for, you know, processing of these sensory stimuli. So at this point, we patented our ideas and we moved on to the next step of actually what does this look like clinically. So one of the things that I guess could have been a criticism or could have led to sort of a confound is how slowly we delivered the anaesthesia. You know, we took what happens clinically in 10 seconds and we spread it out over 50 odd minutes. So what we did is we went into lots of different clinical data sets, about 400 of them, and we looked to see if we could identify this plateau in slow wave activity. So looking at where if we could see a plateau with concentration. And what we found in all these data sets was actually, yes, we could see it in induction in the majority of cases. And it occurred for lots of different anesthetic agents. And it occurred in the presence of other general anesthesia drugs, such as painkillers, opioids, and muscle relaxants. And what this data suggested to us was that if indeed SWOS is perception loss, if this is where our processing of sensory stimuli alters under anesthesia, that maybe 50% of individuals will receive double the anesthesia they need, and maybe 20% won't achieve full unconsciousness. Another thing that we did by then looking at our propofol data was we compared induction and emergence. And these aren't necessarily symmetric processes. You know, we don't necessarily wake up from anesthesia in the same way we go off to sleep. So what you can see here is the slow wave activity fits against concentration for induction in red and emergence in blue. And what we found was actually there seemed to be two separate types of signatures here. We had some people who had a very symmetric induction and emergence from anesthesia, whereas some people seemed to get stuck in this state and had an abrupt change in their slow wave activity when they wake up from anesthesia. So this is where they recovered responsiveness. So they seem to get stuck in the state and only come out of it as soon as they recovered responsiveness. And what we found was that when we looked at all these patient data, was a third of individuals get stuck clinically in this state. And those people who do get stuck are more likely to wake up confused and suffer from delirium. And it may be that this might relate to a certain degree of over anesthesia in the data. So my recent work has really focused on now trying to see if we can titrate anesthesia in real time to achieve this EEG endpoint. And we put a system together that sort of does this dynamic modeling of SWOS, so looking at these changes of slow activity with concentration and providing some sort of element of visualization for the anaesthetist so that they can tell whether they need to increase the dose. And here we can see the slow waves are increasing and the model tells the anaesthetist, okay, you're about there now, you can stop delivering anesthesia. And we've taken this system and we've been applying it in two different studies. And the whole idea of this is that we want to minimize this tail. We think this is the hotspot where you've just lost perception under anesthesia. And anything you have in excess of that is over anesthesia. So we've been using this both in a pre-surgery study, and also we've been doing another EEG MRI study where we've been titrating sevoflurane anesthesia. And in both of these cases, situations, experiments, we've been using certain readouts to try and understand whether SWOS does equal perception loss. And Olivia mentioned this earlier on, we've been using the modified isolated forearm test. So put simply, in our context, we ask the patients, basically, we ask them to open and close their hand under anesthesia. And scarily, in a study performed by one of my colleagues, Jamie Slay, he found that at clinical levels of anesthesia during surgery, 5% of individuals would actually respond to that request. We're also using pain stimuli again. So in this case, we're using a heat stimulus to try and use that as a readout to see if the brain is responsive to pain when held at our endpoint. So I had some preliminary results, and what we found so far is that actually when, both in the patient study and in the healthy volunteer study, when people have anaesthesia titrated to SWOS, we have had no instances of positive responses to the isolated forearm test. The imaging data all looks very good as well, that in response to, if we look at the brain activity in response to the command to perform the isolated forearm test and also to the pain stimulus, is that actually in comparison we see no activity at SWOS compared to wakefulness. So these are all, you know, great supporting evidence that SWOS may be a good marker of perception loss under anaesthesia. So my next step now is we're really trying to now take this forward and do a much bigger study where we try and confirm clinical effectiveness. So we're, due to the current times, we're having to move this study to New Zealand, but we want to try and do this study in about 200 patients now, but also look at other outcomes such as, is there any awareness retrospectively, and do our patients actually wake up better when they have the maybe anaesthesia delivered to the optimum level as defined by SWOS. Okay, so I'm not sure, Andre, do I have time or would you like me to finish there? Should I just do this last little bit really quickly?
Yeah, you can finish up.
Okay, so a key question, and I guess this goes back to Johann's introduction right at the beginning, is okay, so if we think SWOS is perception loss, what is actually lost at loss of responsiveness? Why are individuals no longer willing to engage with us? And so using the same data, we went back and we did a sort of a targeted analysis looking at loss of responsiveness, and particularly looking at the brain's response to stimuli around that time. So it's been postulated that there's a region of the cortex rather than something sort of lower down in the thalamus or brainstem that underlies loss of responsiveness. And we hypothesized, you know, going back to Johannes's empirical measures of consciousness, we hypothesized that if there was a cortical gate, it would have reduced response to all sensory stimuli across this transition. And here are the results if you look at them. So this is what's active in the brain in response to pain, to words and to tones, just before people become unresponsive. And this is the activity just afterwards. And I hope what you can see is pretty much everything in the brain is still responding. We think they lost responsiveness to tone slightly earlier than the other ones because these were less salient. But in general, a lot of these sensory networks are still receiving and processing information. So what we did is we did a subtraction. We looked at what was more active here than here. And then we said, okay, so, and if we do a conjunction analysis, we can look at what's commonly lost to all of these. So these were the regions that had altered activity around the transition. And there was only one region of the brain that in the conjunction analysis that showed reduced activity to all stimuli around this point of loss of responsiveness. And this was this region of the dorsal anterior insula. And what was interesting was around the same time, there had been an experiment where there'd been electrical stimulation of a region very similar to ours. And they'd found that actually if an individual was performing a motor tapping test, that actually stimulation of this region in an epilepsy patient would stop, that responsiveness would stop the tapping. And what we know about the anterior insula is that it's very much, it's a key part of sort of the salience and threat detection network. And it has been postulated to be the site of the sentient cell. So if we're talking about philosophy and bringing in philosophy, and our meta-analyses, you know, various imaging data show that the region that we'd identified was in the cognitive domain. And one of the things that we put out in this paper was actually, you know, we think of the anterior insula as a multisensory integrator, and we know that it seems from the imaging that the stimuli is still being processed, but it might be that this reduction in this region reduces the link to self. So it's maybe so they may still be feeling and perceiving the stimuli, but they no longer link them to them. Okay, so I'll just wrap up now. Hopefully I've convinced you that multimodal neuroimaging is a powerful tool to investigate unconsciousness under anaesthesia. And, you know, potentially slow activity saturation may be a good marker to move forward in terms of assessing depth of anaesthesia and optimizing drug dosing on an individual basis. And it does seem to, you know, the previous work, it does seem to have a sound neurological basis, and we found it has the alterations with age, etc., that one would expect. We've also shown it occurs both during clinical induction and emergence from anaesthesia, and we now have, you know, shown we have the ability to titrate anaesthesia to achieve and maintain this state as well. So we're looking forward to the next stage where we do surgery at SWAS. I'd just like to thank all our funders and students and collaborators who've contributed to this work, and thank you all for your attention.